Hello and welcome to Biotech Studio. In this video I will show you how replication takes place in eukaryotes. DNA replication is a process by which DNA molecule makes copies of itself. The process takes place before cell division. Before we actually talk about DNA replication, we should revise little bit about DNA structure. So in this figure we can see that DNA is a double-stranded molecule. The two strands of the DNA runs in opposite direction, and the two strands are held together by hydrogen bonds. The third important point to keep in mind is that DNA has complementary base pairing. So by the term complementary base pairing we mean that adenine will always base pair with thymine and cytosine will always base pair with guanine. Now look at this sequence of nucleotides, which is AGCTTCA. So on basis of complementary base pairing we can easily tell the sequence of the other strand of the DNA molecule. This complementary base pairing is really important for DNA replication to take place accurately. Now let's talk about steps of replication. DNA replication is divided into three main stages, namely initiation, elongation, and termination. The enzymes required for each step of replication are shown here. Now let's see what happens during initiation. As we know that eukaryotic DNA is bound to proteins known as histones, so before the process can actually begin it is important to make DNA accessible to the enzymes and proteins of replication. So there are certain points on DNA where replication can begin, and these points are known as origin of replication. At early stages of DNA replication, certain protein recognize the origin of replication and then recruit other proteins to the site. Two copies of an enzyme called helicases are among the proteins recruited at the origin. As two helicases binds at the origin, it leads to the formation of a replication fork. As the two strands are held together by hydrogen bonds, the function of helicase is to break them. As soon as the helicase breaks the hydrogen bonds between the two strands, another enzyme called topoisomerase gets activated. Topoisomerase plays role to reduce the strain. Single-strand binding proteins attach to the open strands to prevent recoiling. The two separated strands now act as templates. Another enzyme called primase introduces primer, made up of RNA. The introduction of primer marks the beginning of second stage of replication known as elongation. After that, DNA polymerase III adds nucleotides to the growing end of the chain. As the two strands of DNA runs in opposite direction, the replication takes place differently on each of them. It is because of the limitation of polymerase III, which can only add nucleotides from 5' to 3' direction. This strand is known as leading strand. However, a strand is synthesized in parts in front of strand with 5' to 3' polarity. This strand is known as lagging strand, leading to the formation of small fragments known as Okazaki fragments. The synthesis then continues, leading to the formation of two strands. Once the elongation is completed, the last step of replication, that is, termination, begins. Flap endonuclease 1 marks the start of this step. These enzymes work to remove the RNA primer portion of the newly synthesized strand. Once the primers are removed, DNA poly 1 gets activated to fill the gaps and synthesize the missing part. Finally, ligase enzyme makes the sugar phosphate bonds between Okazaki fragments to fill the gap. This way a DNA replicates in eukaryotes. Hope you like this video. Thanks for watching.